Progress. The Balik Scientist Act or Republic Act 11035 was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik scientists will have an easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik scientist is to be not just a teacher or a facilitator, but also a pusher of innovations within the program.
Balik scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik Scientist Program, is tasked to facilitate the placement of the Balik Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik Scientist Program. Kay being one, I really truly felt that Balik scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
We are now living in a new normal. This COVID-19 pandemic marks as one of the global challenges experienced in this generation. It forces every sector of our society to innovate in order to move forward. We, at the Industrial Technology Development Institute of the Department of Science and Technology, is trying every possible ways to continue our service to our people without compromising the safety of each and everyone.
tracing its roots back in 1901 from the then Bureau of Government Laboratories, which became the Bureau of Science, the Department of Science and Technology, Industrial Technology Development Institute, or DOST-ITDI, turned 119 years old on July 1, 2020. From basic researches to mapping of the country's flora and fauna and other local resources for scientific studies, DOST-ITDI has been a vital instrument in establishing the research and development agenda in the country. It was 1958 when the Bureau of Science became the National Institute of Science and Technology, NIST, that industrial R&D started gaining ground while harnessing local resources and skills towards self-sufficiency and optimized productivity. Industrial R&D went full gear in 1987 when the NIST was renamed Industrial Technology Development Institute, focusing on 4 major functions: research and development, technical services, technology transfer, and custodian of the National Unit of Measure to provide international traceability. DOST-ITDI research and development covers 5 major areas: food, environment and biotechnology, chemicals and energy, material science, and packaging technology, all aimed at supporting and answering the needs of local industries. Complementing its R&D are its technical services, standards and testing, national metrology, and technology transfer, aimed at harnessing local industries' productivity and competitiveness, and translation of developed knowledge or innovation into the production sector, paving the way for new businesses or startups. As well, DOST-ITDI innovations serve as springboards for businesses to thrive and prosper. In support of the administration's thrust in addressing the COVID-19 pandemic, DOST interventions are anchored on the theme, Agham at Teknolohiya, Sandigan ng Kalusugan, Kabuhayan, Kaayusan, at Kinabukasan. At the height of the COVID-19 pandemic, and when most of the country was in enhanced community quarantine, DOST-ITDI bravely rose to the call of duty, distributed ready-to-eat foods such as the Pack of Hope and Mung Bean Cocoa Milk Drink to our frontliners in Metro Manila and other regions in the country, produced face shields via 3D printing and donated these to hospital frontliners, developing prototypes and 3D printing critically important parts of hospital equipment, and improved design of N95 mask to better protect the frontliners. The Institute is also providing interventions for our displaced countrymen who lost their jobs and livelihood by making training available online whenever necessary. And even before this pandemic, DOST-ITDI innovations were critical in rehabilitating communities that experience calamities and even war and make them whole again. DOST-ITDI has been preparing for an innovative ecosystem for new knowledge and technology to thrive and help make us ready for Industry 4.0. DOST-ITDI aims to achieve kaayusan and ascertain the future or kinabukasan through its initiatives and help businesses in every Filipino adapt to COVID-19 under the new normal. State-of-the-art facilities are being established. Construction of the Simulation Packaging Testing Laboratory, SPTL, and Green Packaging Laboratory, GPL, is ongoing. At the SPTL, stress conditions that affect products during transport are simulated that can help mitigate losses during distribution, while produced products can be processed and packed in a green packaging laboratory. AMSIN, or the Advanced Manufacturing Center, DOST's 3D Printing Technology Center, is a joint project with Metals Industry Research and Development Center, MIRDC. ITDI focuses on developing multiple 3D printing materials from local materials to reduce cost. Halal Food Research and Development Facility. With this facility in place, the Institute hopes to develop new food products that are compliant to halal standards and as well support DOST as it responds to Republic Act No. 10817, or the Philippine Halal Export Development and Promotion Act. Enhancement of the competence and capabilities of the National Metrology Laboratory of the Philippines. Expertise and facilities are being upgraded, and construction of laboratory facilities for metrology and chemistry and biology are now ongoing. It is envisioned that the NML will provide the country with credible measurements and traceability in the fields of physical, chemical, and biological metrology. And with the emergence of the COVID-19 pandemic, DOST response has been decisive. With the support of President Duterte and the Interagency Task Force for the Management of Emerging Infectious Diseases, the DOST will establish the Virology Science and Technology Institute of the Philippines, or VIP, to be constructed at the Nucleic Economic Zone in Capas, Tarlac. The VIP shall be pursuing priority virology research and developing diagnostic kits, therapeutics, and vaccines for diseases caused by viruses, where DOST-ITDI will have a critical function.
From laying the groundworks for science and technology in the country, the Industrial Technology Development Institute of the Department of Science and Technology, through the years, which turned 119 last July, has been consistently providing innovations to industry to help make them competitive, emerging as a credible industry partner. The Institute has been instrumental as well in mitigating hazards and improving the lives of disaster victims and communities to rise again. With so much optimism with this cooperation and bridging of talents and expertise, we look forward to enhance science, technology, innovation, competitiveness, and the emergence of new research and development capabilities that hopefully will translate into new products and services that meet the current and future needs of our nation and the people.
All right. That was our video about the VIP program and also the briefer for the DOST Balik Scientist program. Again, welcome to all our learners. We now have 116 participants here on Zoom, while we have 35 on Facebook and 4 on our YouTube Live. I can see that we have participants from CSU, CVSU, RTU, PLM, UP Diliman, also Quirino, Cagayan, Laguna, and others. I think all over the Philippines. And I guess all are excited to learn about diagnostic virology. But before we start today's session, let's have first a recap of the previous webinar. Last Friday, February 18, Dr. Christina Layson, one of our Balik scientists from the USA, gave us an introduction to the molecular virology, genome sequencing, and bioinformatics. There, she gave us a briefer of what the viruses are and how do we classify them. She also mentioned the sequencing technologies that we currently have and lectured about evolution and phylogenetics. And then lastly, she helped us apply these topics about sequencing and evolution and phylogenetics in looking how we understand SARS-CoV-2 lineages. For today's webinar, we shall be learning a lot about diagnostic virology. But before we proceed to the meat of our session, let me formally introduce our distinguished Balik scientist and resource speaker this morning. Our speaker obtained his bachelor's degree in biology and microbiology at the University of the Philippines, Los Baños. After taking his master's in microbiology, doctorate in microbiology and animal virology, and postdoctorate in aquatic virology at the University of Hawaii at Manoa, Honolulu, he pursued postdoctorate in medical biotechnology at the National University of Singapore, then another postdoctorate degree in aquatic virology at the University of Hawaii at Manoa, and finally a postdoctorate in medical biotechnology at the University of Cambridge in Cambridge, United Kingdom. He is a virologist and microbiologist with 20 years of experience in academic research and 17 years in the industry developing diagnostic assays for the detection of bacterial and viral pathogens. He co-founded Diagnostics for the Real World Limited in 2003, where he led the development of several rapid diagnostic tests for the detection of hepatitis B virus surface antigen, human immunodeficiency virus antibodies, and hepatitis C virus antibodies, as well as improved versions of the chlamydia rapid test. He also worked in the food and environmental testing industry from 2008 to 2018, where he led the development and production of PCR and qPCR agents and diagnostic kits to detect pathogens, parasites, toxins, GMO, antibiotics, allergens, and other adulterants in food and water. So now let's give a warm welcome and a big virtual round of applause to Dr. Elpidio Cesar B. Nadala Jr. Dr. Nadala, the virtual floor is yours.
Thank you. Let me share my screen first. Okay, I hope you see the screen now. Welcome to today's webinar on diagnostic virology. Diagnostic virology, well, diagnosis of a viral infection is the process of determining which virus explains a person's signs and symptoms. The information required for diagnosis is typically collected from a history and physical examination of the person seeking medical care. Often, diagnostic tests are done during this process. And in this seminar, today's seminar, we will focus on these diagnostic tests. Initially, we're going to discuss the characteristics and classification of viruses, and then describe some of the clinical manifestations of viral infections. These are the signs and symptoms of disease. Then the methods used for laboratory diagnosis of viral infections, which include viral culture, antigen detection, nucleic acid detection, and serology. So what are viruses? They are extremely small, obligate intracellular parasites comprising of genetic material like DNA or RNA, often surrounded by a protein coat and sometimes by a membrane. All viruses have two things in common. Each is encases its genome in a protein-based shell, and each virus relies on its host. That's why they're obligate intracellular parasites. So they rely on their host to reproduce. And but beyond that, these commonalities, there lie endless variations. As you can see, the virus can be very small or it can be large in all different shapes. Viruses are highly evolved life forms that are continuing to evolve. So they're as old as the oldest cell, living thing. So they have been evolving since life started on Earth. And so they are quite old. There are just when and how were viruses discovered? Well, early in the last century, the first human virus, yellow fever virus, was discovered as a filterable agent of disease. And after that, a series of other viruses, rabies, variola, which is a smallpox virus, chicken leukemia virus, polio, Rouse sarcoma, so all at the early 1900s. And finally, influenza, of course. They were all discovered as filterable agents of disease. Filterable meaning they pass through very small, tiny filter holes in the filter, which would normally retain bacteria and other microbes. So there are just 9,110 named species of viruses that's listed by the International Committee on Taxonomy of Viruses. Officially classifying a virus used to require scientists to culture the virus in its host. As we said, a virus needs a host to replicate, or host cells. It's very time consuming and, if not, an impossible process sometimes. So in addition, identification has been biased towards viruses that cause disease in humans or organisms we care about, such as farm animals and crop plants. Of course, viruses that do not normally cause disease or do not overtly cause disease in these animals or humans, we never really studied them. Over the past 10 years, the number of known and named viruses has exploded because of advances in technology for finding them. And we'll talk about what these are, plus a recent change to the rules for identifying new species, to allowing naming without having to culture the virus in its host. So there are 6 realms of virus, and the biggest one, of course, are the viruses that use an RNA-directed enzyme, and this includes SARS-CoV-2 and the other coronaviruses. Then there are the DNA viruses, double-stranded DNA viruses with a common shell structure. Example is the human herpes viruses. Then there are single-stranded DNA viruses, and these include the papilloma viruses. And the rest are quite small. And so these are the major ones, including, of course, the Viridnaviria group, which includes the African swine fever. So they come in all shapes and sizes. You have the giant Mimi virus, which is same size as bacteria. You have the lunar lander-shaped virus, bacteriophages actually, that infect bacteria. And of course, you have the usual icosahedral-shaped viruses. You have the filamentous viruses and the bullet-shaped rhabdoviruses that cause, for example, rabies. And the pox virus is a relatively big one. So under the electron microscope, you would see these different shapes and sizes of virus. Scientists estimate that there are about 10 to the 31 individual viral particles inhabiting the oceans alone at any given time. That's 10 billion times the estimated number of stars in the known universe. So hard to imagine how many virus particles there are. So we live and prosper in a cloud of viruses. A lot of viruses surround us all the time. But most of these encounters have no consequence. Many, though, are inapparent or asymptomatic infections. Some viruses cause disease, in fact, and over 60% of infectious disease cases seen by a physician, the doctors, are due to viral infections. And viral infections of the lower respiratory tract, that means your lungs, and diarrheal diseases are among the leading causes of death. And if you look at this graph, for example, lower respiratory infections, 2.5 million deaths in 2017, diarrheal disease, over 1 million, and HIV/AIDS, of course, almost 1 million. So how is influenza, for example, diagnosed? Well, the doctor will see influenza-like illness, fever, at least 100 degrees Fahrenheit, cough or sore throat, and no other known cause. So that will have to undergo rapid tests and then confirmed by PCR and viral culture and serology. So that's kind of the process for diagnosis. For COVID-19, the infection affects several body systems, including cardiovascular, gastrointestinal, nervous system. Although it starts off as an upper respiratory tract infection, like your sinuses, your pharynx, larynx, If it progresses to lower respiratory tract, like go down into your lungs, then they become systemic, get into your bloodstream and spread to your vital organs, causing all of these different signs and symptoms of disease. Some viruses cause skin rashes in humans. So they're manifested as rashes. And for example, measles, smallpox, chickenpox, and others, including rubella, which is German measles. The immunopathology, the clinical signs and symptoms of viral infections or viral disease are mainly immunopathologic in nature. That means your own immune system causing damage to your body. And damage can be either T cell mediated or B cells. You've heard about this recently, of course, B cells being antibody based, and these ones are cell mediated based, which are lymphocytes in our body that attack virus infected cells and kill them to stop the virus infection. So among the damage caused by T cells are infections of like hepatitis B, HIV-1, herpes, and also RSV, respiratory syncytial virus, which is another respiratory disease, mainly in kids. Now, the B cell mediated, antibody mediated damage is a very good example is dengue virus, which we experience in the Philippines. Now, there are several general patterns of virus infection. One is acute. This happens with common cold virus like rhinovirus, rotavirus, which is a diarrheal type, enteric virus, mostly in kids and young children, and then of course influenza. So the pattern is that when you get infected, virus gets produced, you get symptoms, and then virus goes away, and that's it. That's acute infection. For latent infection, after initial infection, the virus titer go up, and then you get symptoms, but then it disappears. It may show up again at the later stage with symptoms, or it shows up without symptoms, or at the late stage of your life, you might get it like that. So that chickenpox is, for example, one of these diseases where older people can actually start producing virus again and have really painful symptoms, lesions in the skin. Then you have the third type, which is persistent. You have no symptoms, but virus is continually produced throughout life. And lastly, you have persistent pathogenic. That means you get this acute infection early, then you have a small amount of virus being produced, no symptoms, but then at some point later, virus is produced again and actually kills you. Okay, so that's like HIV, where you get initial infection and you carry the virus. At some point later on, it will actually kill you. So that's a persistent pathogenic virus. So indications. So what are the main reasons why we need to do diagnostic virology or need to identify viral infection? One is for proper management of certain diseases. For example, if rubella or German measles is diagnosed in the first trimester of pregnancy, abortion is recommended. Or if a pregnant mother is HBs, hepatitis B surface antigen positive, abortion is recommended. Second reason is diagnosis of disease caused by viruses, for which antiviral treatment is available. So in the case of herpes, for example, You get the zoster symptoms later in life, which is very painful. So that can be mitigated by an antiviral against herpes. Same with SARS-CoV-2 now, we have Paxlovid and molnupiravir. So you want to be diagnosed early, then you can take the antivirals. Third one is screening of blood donors. So we have blood banks. We want to make sure that hepatitis B or HIV or other bloodborne viruses are not in the blood, so that these have to be tested. And so you prevent the spread of the virus. Fourth is early detection of epidemics and pandemics. So in the case of SARS-CoV-2, for example, if we have detected it early and prevent people from spreading it, then you could control it. Okay, so those are the main reasons why we do diagnosis, identify a viral infection. And so how did we classically demonstrate a viral infection? It's through this, what we call reverse postulates. First, we isolate the virus from the host. Okay, so you have a rash, for example, and you have vesicles in your skin, so we could get a biopsy from there, get the virus, then grow it in host cells. So these are cell cultures that you can introduce the virus, and the virus, if it's the right host, will grow. And then whatever you get there, you need to filter that and show that it's smaller than a bacteria. Okay. And after that, you need to reintroduce what you grew into the experimental animals to show that it can reinfect. Okay, because if it's the original host, then it should be able to infect another host. Then you isolate the virus from that infected animal and then detect it using like antibodies that are specific for the virus. That's just the classical way we did it. And what are the approaches now for lab diagnosis of viral infection? Number one, you have to demonstrate the virus and its components, okay, that they are there, that they're present in the biopsy or in the sample. Then you isolate the virus, probably grow it in cell culture, and detect with specific antibodies. So this is kind of a summary of a shortcut of the reverse postulates. So how do people isolate the virus, or how did we traditionally isolate the virus? You can use lab animals, for example, mice. Many viruses can be grown in mice. You can also use fertilized hen's egg. So different parts of the egg, the fertilized egg, that means there's a growing chicken there. Different parts of this egg can grow different viruses. Okay. And then organ tissue or cell culture, now of course mostly cell culture, and we'll talk more about this later on. And then once it's grown, then you can identify the virus by serological method. That is, if you have antibodies specific for the virus. There are other ways to identify the virus that you have grown. Nowadays, it's more common to use cell culture to grow virus. So what are the biological specimens that are used in diagnostic virology? If it's a respiratory tract infection, for example, SARS-CoV-2, so you take throat and nasal swabs, nasal and bronchial washings, or sputum. If it's an eye infection, you take throat and conjunctival swab, conjunctival of your eye, or scraping. And if it's gastrointestinal tract infections, then you take stool and rectal swabs. For vesicular rash, that's rash with vesicles on your skin, then you take the fluid or skin scrapings. And if it's a maculopapular rash, which that means doesn't have fluid or anything, you take throat, stool, or rectal swabs. Now, for viral encephalitis and meningitis, these are infections of the central nervous system. You take stool, tissue, saliva, brain biopsy, or cerebrospinal fluid. For genital infections, you take vesicle fluid or swab. And for urinary tract infections, urine samples, and for bloodborne infections, blood, of course. So, viral culture. Well, back in 1949, scientists were able to grow poliovirus in cultured cells. Other viruses have also been cultured, discovered, and vaccines developed for them, for example, measles and rubella. Cells in culture are still the most commonly used hosts for propagation of animal viruses. And before quantitative methods for measuring viruses were developed, evidence of viral propagation was obtained by visual inspection of cells, the infected cells. So what are the cell types used? Well, we have primary cells. These are cells taken right out of the tissue of the animal, for example, a monkey. So they would harvest the kidney and release the cells from the tissues and use that for culturing. Some viruses require primary cells for growing. HAV, for example, before hepatitis A virus, used to require primary cells. And I actually did some of this culturing. There are semi-continuous cells, but primary cells will die off, okay? They won't divide too much. But semi-continuous cells can divide, can undergo many divisions before they die off. And examples of these are human embryonic kidney or skin fibroblasts. But there are cell lines that are continuous cells. That means they keep on dividing and you can keep on growing them. And these are like the HeLa cells, which is the cervical cancer cells from Henrietta Lacks, Vero cells, which are African green monkey kidney epithelial cells. HEP2 cells are actually derived from HeLa and others, made from like canine kidney cells. It depends on what virus you want to grow. Now, viral culture of SARS-CoV-2. SARS-CoV-2 can be cultivated in various cell lines that express the ACE2 receptor, angiotensin converting enzyme 2 receptor, which is required for viral entry. So viruses will only grow on the host if it can enter the cells of the host. So that's why receptors are very important for viruses. The monkey kidney cell lines, Vero, CCL81, and E6 are the most used for SARS-CoV-2. And CPE is often observed within 3 days of inoculating these cell lines with SARS-CoV. And subsequent analysis of culture material by real-time PCR or immunofluorescence staining can be used to confirm presence of SARS-CoV-2. Of course, right now, this is more popular now than doing any staining. Recovery of SARS-CoV-2 in viral culture has served as the reference standard for detecting replication competent or infectious virus throughout the COVID-19 pandemic. So in viral culture, for example, we look for cytopathic effects that indicates the presence of the virus, replicating virus. So if you had a monolayer of cells growing nicely here, very healthy, once you infect them, after a few hours, you start seeing cells that round up and show the cytopathic effect. And after more time, maybe overnight, 24 hours, all of the cells would have come off from the monolayer and lysed. There's another type of cytopathic effect besides just rounding up and detachment. And it is these syncytia formation. These are giant cells that form from fusion of several of these cells that they fuse together and become giant cells. That's another cytopathic effect of some viruses, mostly the envelope viruses. So this so-called cytopathic effect or rounding up of cells, they cause plaques or clear areas in the monolayer of cells to form. So it's a good indicator for individual virus growth. And so they have used that for calculating virus titer. Example, you have a sample where you don't know how much virus you have. You just dilute that and make many dilutions, and then you plate out those dilutions. And if you count the plaques, then you know how many viruses or infectious virus particles you had in your sample. So viruses are very, very small. If this is an animal cell, for example, which would be, let's say, around 10 microns, 10 to 20 microns. Virus is this small. So it will be less than 1 micron. And it's a lot, even it's smaller than bacteria. So you really need, most of the time, you will need an electron microscope or a cryo-electron microscope to see the virus. And that's actually one of the ways that they are able to identify the virus. Okay, again, electron micrograph, bacteriophage, influenza virus, rabies, and polio. They all. So if you can see them on the microscope, then you know which virus they are. Serology. Well, the specificity of the antibody-antigen reaction, you know, everybody heard of antibodies. Well, antigen is just a protein, a shape on the surface of the virus or even inside the virus, a shape of a molecule that makes up the virus. This specificity is used to design a variety of assays for viral proteins and antiviral antibodies. Okay, as we said before, when you do diagnostic test, you either look for viral protein or viral antiviral antibody or nucleic acid, the genome of the virus. So the techniques that are based on viral proteins and antibodies include immunochromatographic assay, which is just lateral flow or dipstick assay. People have seen or used pregnancy kits or tests. That's what lateral flow is. And of course, the rapid antigen test that you guys probably have seen, that's also lateral flow. But there's another assay called the enzyme-linked immunosorbent assay, or ELISA for short. But these are done in the lab with equipment. Then you have immunofluorescence staining, which is not very popular anymore. But it used to be very, very popular because of use of specific antibodies, and then you could see the stained cells. Now, so example of the lateral flow assay. This is looking for antibody against COVID-19. So this would be your device, the rapid antigen test device. And normally, if you're looking for antibody, then you take a blood sample, put that in this particular sample well here, and then you just basically chase it or push it in with buffer, just the liquid. And then as you incubate it, the liquid travels up this membrane here, which is made up of nitrocellulose. So but within the device, you have gold labeled antigens. So in this case, maybe a spike protein that's attached to gold particles. We call nanogold because they're very small. They're about 30 nanometers in size. 30 nanometers in size. So about the size actually of a virus particle. And then you have the antibodies, of course, that came in with the blood. So if they have antibodies to SARS-CoV-2, that antibody will bind to the antigen of, or the spike protein of SARS-CoV. And so you get these two together, they go in here, and if you have anti-human antibodies on the test line here, as these things pass by, they get captured. And if the if the antibodies attached to a virus antigen that's attached to gold, then you see a red line, just like illustrated here. And additionally, if some of these antibodies do not actually bind here, and then they can get caught in the next line, which is the control line, which is antibodies against whatever this antibody is. It could be, well, this is human antibody, of course, but if you had put in as well rabbit antibody that's labeled with gold, then that would be captured in the next line. The rabbit antibody, of course, will not be captured here because this is anti-human and the C is anti-rabbit. So the examples of results from a lateral flow assay, if it's positive, you have the control line and the test line. If it's negative, you only have the control line because these guys will be captured here, but these ones are not going to be captured because you don't have, or they're going to be captured, but they don't have any gold particles on them. So you won't see any line. And if you don't have the control line, then something went wrong. And they call that invalid, and they have to test you again. So for a rapid antigen test, the sample is different. It's not blood. So they take like a nasal swab. This one is a nasopharyngeal swab because it goes all the way to your pharynx. Some just nasal swab will just go to the your nose. This one is deep. And then it's going to be put into a lysis buffer, or a sample buffer. And then in this case, the case of this test, they put on this cap and then put drops in, which now will contain, if you have SARS-CoV-2, SARS-CoV antigens, and then it develops into the test and control lines if you're positive, and only a control line if you're negative, and no control line if the test is invalid. Now, what's going on here? So it's quite different from the antibody test. And what's different is your sample has the viral proteins, let's say spike proteins, and you have antibodies that are labeled with gold this time, not the antigen, but the antibody is labeled. And as the sample passes through this conjugate pad, it gets caught, or the antibodies bind to the antigen of the SARS-CoV-2. And you have another set of antibodies on the line that can bind also the same antigen. So you get this sandwich, and you get the signal from the gold that's attached to the antibody. Okay. If there is no SARS-CoV-2 antigen in the sample, then you won't be able to get this and you won't have a line. Okay, and that's why it's negative. But then you also have, in the control line, you have antibodies that recognize these antibodies. Like if they were made in rabbits or whatever, then when the antibodies pass through here, whether they have the antigen or not, they will form a line. And that's why it's a control line. Okay, so that's antigen rapid test. Now, the thing with rapid tests is that you've heard that they're not as sensitive as RT-PCR tests because they require a lot more virus in the sample, or virus antigen in the sample, to be positive. Okay, so that's why if you're positive with antigen tests, that means you have high amount of virus, high enough so that it's positive. But if you're negative, doesn't mean you don't have the virus. It just means that you don't have enough virus in the sample that was collected from your nose in the swab sample. So it's known that rapid antigen tests are able to detect, as a sensitivity at best, 83,000 copies per mil, and most of them actually require 1 million copies per mil of sample before they be positive. Compare that with RT-PCR, where most of the RT-PCR detect just 1,000 copies, or in the case of our swab test, it can detect 500 copies per mil. So you're like almost 1,000 times more sensitive than a rapid antigen test. So the other format, as I mentioned before, is enzyme-linked immunosorbent assay, or ELISA. And this is done in the labs with liquid reagents. And there are two formats I'm showing here. The straightforward formats. There are other formats for ELISA, but if you're looking for antigen, for example, normally you would coat your plate with the antibody, because then you will add your sample with antigen and then follow that with another reagent that has an indicator, like an enzyme attached to the antibody. So you form the sandwich, but instead of like gold particle to show, to reveal the positive result, you have like an enzyme that will turn the color of the liquid into blue or yellow or orange, and then you know that you have a positive result. If you're looking for antibody, then you would coat the viral antigen on your plate, and then you follow that with your sample. If it binds to your antigen, then you follow that with another antibody that will contain the enzyme. So only if it binds will you see signal, because in between adding antigen or antibody, you're gonna wash the plate. Okay, so this is how it looks. Yeah, you got wells. Normally it's a plate with many wells, 96 wells to be precise. And so each well, you would have a coat of antigen if you're looking for antibodies, and you will coat with antibody if you're looking for antigen, right? And in between addition, you have to wash the well until you add your substrate, which will turn a different color if you have a positive result. Yeah, if you have enough. So like here, you get all these yellow for positive wells where your sample was either you detected antigen or you detected antibody, depending on your reagents. So as I said, there are various ELISA formats. You can have a direct one here, an indirect one, and an even more amplification. So they have several antibodies before you get your signal. And you also have competitive ELISA, where you compete with. So if you're looking for like a protein, an antibody that binds this, you will have to compete with this antibody that you already add as your regular reagent. So that's ELISA. And I have been mentioning antigen detection by immunofluorescence assay. Again, it's using antibodies, but this time your label is not gold, it's not enzyme, but it's a fluorescent dye, yeah, like FITC. So if you're looking for viral antigen, you would stain with antibody that has this dye. And if your antibody is specific to that antigen, it will bind and label where that antigen is. So if you had a cell, for example, and you're looking for virus, then you're gonna stain that, besides staining for other parts of the cell, with antibody against the virus. And if your antibody is labeled with a green dye, then you will see it where the virus is in the cell. In this case, you also have labels for other parts of the cell. Now, hemagglutination or hemagglutination assay is one of the assays that were used for viruses that have proteins on them that bind red blood cells or erythrocytes. So some members of the adenovirus group, the orthomyxoviridae, where the influenza virus group are, or the parainfluenza virus, they have proteins on the surface that are able to bind red blood cells like this. So when these are like the virus, and so they stick to the surface of the red blood cell, and they create a network. Yeah, they fuse different cells together. So, for example, here, if you put red blood cells in each of these wells, and then you add a serial dilution of the influenza virus. So when they agglutinate the red blood cell doesn't gather at the bottom, because this is like a V-shaped well. So without agglutination, you see a button, like a distinct button at the bottom. But if it agglutinates the red blood cells, then you won't see that gathering there, because they form a network of aggregated erythrocytes. So the more dilute, the more likely you're not going to be able to form agglutination, but at lower dilutions you will. Here you have so much virus that the neuraminidase on the surface of the virus is starting to break the agglutination apart, but that's a different phenomenon. So nucleic acid detection, finally to the point where diagnostic virology is at this time. Just a few years, well, a couple decades ago, these methods for diagnostics were not actually used yet. So these are new. And as you know, PCR is towards, mid 1980s. And so once that was developed, then we started using that for diagnostics. Of course, more recent development is next generation sequencing, where you actually are able to sequence the genome of the virus. And these are both replacing cell culture as the method of choice for virus detection. Next generation also sequencing is useful for discovery of new viruses or virus variants, especially those that are associated with human disease. So you hear about sequencing for, Omicron or Delta virus or different variants for SARS-CoV-2. So detection of nucleic acid. So as an example here, I have detection of SARS-CoV-2 RNA, and the most common and popular method is RT-PCR, or reverse transcription polymerase chain reaction. And less common is isothermal amplification. And actually, the assay that my company is making against SARS-CoV-2 is based on NASBA. And another company called Hologic uses transcription-mediated amplification, which is similar to NASBA. And Abbott, if some of you may have seen it, the Abbott ID Now assay uses recombinase polymerase amplification. Now, these are all very fast compared to RT-PCR, so they are more of the rapid type point-of-care tests. They are ready in less than an hour or less than 2 hours. RT-PCR, you wait many hours, at least maybe 5 or 6 hours before you get the result. Next generation sequencing, of course, that's what we use to sequence. So the process for SARS-CoV-2 RT-PCR, for example, again, you take the nasopharyngeal swab. So you have your specimen in your tube here, and then it goes through an RNA extraction. It could be manual, it could be automated, but normally they use either a membrane of silica or silica beads to purify or pull down the viral RNA and separate it from the rest of the virus. Then that undergoes what they call retrotranscription or reverse transcription. So because the virus is made of RNA, that has to be converted to DNA first, double-stranded, before it's amplified by PCR. And they use different tests use different primers and probes to do the PCR. And if it's real-time PCR, then they look for fluorescence, which increases after several cycles of replication. Each cycle doubles the number of amplicons from PCR. Now, real-time PCR, the one that we talked about where you look for fluorescence, there are two types. One is dye-based. This dye is fluorescent, but it likes to bind double-stranded DNA. So when qPCR starts, of course, there's a separation of the two. This is just PCR, by the way. The reverse transcription is already done, assuming that it's already done at this point. The two strands of DNA are separated. Then you have these primers going on, and then the polymerase synthesizes the other strand from the primers. Then you have two. So each cycle you double. But the role of the fluorophore here, as you generate more and more double-stranded DNA products of the PCR, you get more and more signal, right? So that's the principle of dye-based real-time PCR. Most PCRs are like that because that's cheap, easy, and if you have only one target, you're okay. But if you have multiple targets, sometimes you need to use what they call probe-based real-time PCR. When it's probe-based, you have normally a fluorescent dye attached to your probe, and then a quencher on the other side. The quencher quenches the fluorescence. So it won't fluoresce if it's nearby, it will quench it. But if you cut the probe apart, then this can fluoresce. So what happens during PCR is it's separated, then you get your probe in, then you synthesize. But this Taq polymerase, for example, once it passes by this probe, will also digest it, cut it up, and separate now the quencher from the fluorophore. And as it separates it, it fluoresces. So as you generate or you do more and more of this amplification during each cycle, you generate more and more, you get more and more fluorescence. So it comes out like this. And there are many different types of fluorophores. So you can have 2, 3, 4 different signals coming out from one reaction. That's what probe-based PCR is. Now, NASBA, on the other hand, is isothermal. That means you don't cycle. You just allow the enzymes to work at a constant temperature. And what happens is you have the single-stranded RNA from the virus. So the first probe binds and then synthesizes. The reverse transcriptase synthesizes the DNA part. And then another enzyme called RNase-H digests the RNA, making the DNA single-stranded. And then another primer goes in and makes this a double-stranded DNA molecule that is able to become a substrate for another enzyme, a third enzyme called T7 RNA polymerase, because there's a T7 tail here where it can bind, and it can synthesize many more RNA amplicons. Okay, so even though you lost your RNA here originally, you have many more here that can become a template for making more of this double-stranded DNA and making even more RNA. So these two, they keep going on, and the amplification is very fast. That's why they're used in point-of-care tests. You don't have to cycle the temperature. How do you detect these? One of the methods is by using a molecular beacon. Molecular beacon is like a hairpin type probe where the fluorophore and the quencher are close together. But when they bind to one of these, they get separated far enough so that this fluorophore now fluoresces and you get the signal. Okay. And the other way of detecting it, of course, is by dipstick. So the same process goes on. This is the Samba-2, which is the test that we are producing. My company produces against SARS-CoV-2. And we're using a different device here, but the whole sample preparation is automated. And again, but instead of a graph, we generate a test strip. It's kind of like the lateral flow you saw earlier, where if it's negative, then only one line, the test, but if it's 2 lines or 3 lines, then it's positive. If there's no line or no control line, then it's invalid. That's this test. The other method that I said that Abbott ID now uses is the RPA amplification. It's called recombinase polymerase assay. And it's kind of like PCR, except that you have these recombinase proteins and you don't cycle, okay? The key here is it's isothermal. So you do everything at the constant temperature, say 40 degrees centigrade, lukewarm water, not even boiling or anything like that. So at a constant temperature, if you provide your forward and reverse primer like we do in PCR, but in addition, you have to have recombinase proteins and some single-stranded binding protein and ATP. So this is all driven by ATP. So what happens is the recombinase proteins bind your primer, then actually tries to find the part of your target where the primer can hybridize. So there's specific recognition. Oops. And once that happens, single-stranded binding proteins come in, and then the actual DNA polymerase goes in and displaces the recombinase proteins and starts synthesizing the other strand. So this is essentially like PCR, but there is no cycling. The enzymes continually do this, so it's very rapid. You can have, in PCR you need about 20 minutes, 30 minutes at least. Here you can have signal in 5 minutes, 10 minutes. So this kind of format for amplification and detection is implemented in the Abbott ID Now test, where you have only one cartridge, you put in your swab there, you put it into this device, and it will tell you if within like 30 minutes, it will tell you if you have your RNA positive. Okay, so they claim 5 minutes, but it's really longer than that to get the maximum sensitivity. There's just one thing that people have seen with this device, because it's so simple. Actually, it's not quite as reliable as RT-PCR. That's just the way it is. So those are the nucleic acid tests that most people use. Now, next generation sequencing, we've all heard about sequencing. There's the first generation, and this was Sanger dideoxy synthesis using radioactivity, P32, P33. And you have to run these products in a slab gel and then manually determine the sequence. So it's very laborious, and you can't really sequence a lot. Second generation sequencing was by Ion Torrent and Illumina. These are companies, and Illumina is probably the most popular sequencing platform right now. They have several devices. One that I'm familiar with is the MySeq. But for human genome sequencing, or you need NextSeq or plant sequencing genome. But this is sufficient for bacteria sequencing, and I would think viruses can be sequenced by IC and MiniSeq easily. Third generation is by PacBio, but this device reads each DNA molecule one by one. It reads the sequence one by one, but it does large fragments, thousands of nucleotides it can read in one DNA molecule. But the fourth generation, and to my mind, the most interesting generation of sequencing is nanopore channels. So these are channels within a membrane where you thread, or not you, but the reagents thread the DNA through and determine what the sequence is in parallel. So this is really good technology and very fast. For example, what happens is you have initially, so you have your sample. First, DNA is extracted, and then it's fragmented. This is part of sample preparation. Then a library of the DNA is prepared. So these are all like a kit, that you do manual preparation. So, and then after that, you put it into the device for sequencing. So this library that you've generated during sample prep, you put into the device, and the device starts sequencing it, threading all these DNA molecules through the pores, a nanopore. And as it's determining the sequence, it's already doing an alignment, doing some base calling while it's going through and doing an alignment. And by the time you finish, you can now annotate, meaning look at the sequence that were determined and say, okay, this is what gene this is and that, whatever, or there is a mutation in this nucleotide. And so you could actually find the variants at this point if you were sequencing a virus. And the timeline is you take 2 or 3 hours to do your sample prep, and then the rest of the sequencing another few hours. So typically you finish within the day, like 7 hours, you have a sequence. And this is even for, human genome sequencing. Imagine if you were only sequencing a virus, it would be much shorter. So we can actually, with this kind of sequencing, you can actually determine if somebody has a Omicron variant or Delta variant or whatever in hours, not in days, right? And not in a week, but maybe 6 or 7 hours. Okay. And using this next generation sequencing, the most influential techniques have been called metagenomics. That just means you basically extract DNA from the whole sample and just look at what kind of genome is in there. So you don't have to isolate the virus, in other words. And they've used that in ocean samples, for example. Metatranscriptomics is just basically looking for RNA, metagenomics, DNA. Other technologies, of course, single virus sequencing. That's only to identify if you had a new virus, for example. But there are so many viruses that we haven't sequenced actually, but they exist are all around us. Okay, so in a typical virus infection, for example, you get infected, you have a pre-symptomatic stage. Culturable virus becomes detectable around this point, where the viral RNA is also pretty high. But after the onset of symptoms, it's only when you start seeing positive antigen rapid test. Okay, so at this stage. So onset, maybe 1 week up to 2 weeks, you might be able to see positive antigen. For IgG, it will just start going after 2 weeks. That's why they tell you, if you've been vaccinated, wait 2 weeks, because that's when your antibodies start going up. Similar for IgM, but IgM goes down after like a month. IgG should persist for a while. Okay, so I mean, I'm showing this because we talked about virus isolation, we talked about antigen detection, nucleic acid detection, antibody detection. There are only certain times during virus infection that you are able to detect whatever target you're looking for. And so performance characteristics of diagnostic methods for example, respiratory virus infection, like influenza or SARS-CoV-2. There are many different methods, as we've talked about. There is the nucleic acid tests, like RT-PCR. Sometimes it's manually extracted, or sometimes you have an automated extraction process. Both have good sensitivity and specificity, but the automated one is faster and more user-friendly. Then there's the point-of-care nucleic acid amplification test, like what I said, the Abbott ID Now, or the test that we have, but more like the Abbott ID Now test. Their sensitivity is not quite as good as RT-PCR, and their specificity sometimes also lacking, but they can provide you results within half an hour, and they're very easy to use. Okay. Next generation sequencing, of course, is only used for unknown viruses or determining variants. For antigen detection, the point-of-care antigen rapid tests, they're specific, but the sensitivity is not there. They're easy to use, and the time to result is very fast, like minutes you will see if you're positive or negative, although it's not that reliable. ELISA, of course, has better sensitivity, but it's harder to use, and we've not really used it for SARS-CoV-2 in any meaningful way. And then, fluorescence. Okay, so these different tests have their intended uses. And it depends on where or the setting. For example, in hospital emergency rooms, you want a point-of-care nucleic acid test, because as the patients come in, you don't want to wait 1 day, 2 days before you know what their status is. You want to know within half an hour, or at the most maybe 2 hours, whether they're SARS-CoV-2 positive or not. Then you can admit them or not into your, you know where to put them, whether you want to put them in the COVID-19 wards or in the regular ward if they're negative. Retirement homes is the same thing. It has to be easy to use, and it has to be rapid. In schools, it's the same. So, but for screening, where you have targeted and general population, you can use laboratory-based RT-PCR, which is the predominant one we have in the Philippines. And for variant monitoring, of course, next-generation sequencing. Okay, my last thing about diagnostic virology and what has been the trend. Diagnostic virology is rapidly moving into the mainstream of clinical medicine as a result of the convergence of several independent developments. First, dramatic progress in antiviral therapeutics has increased the need for specific viral diagnosis. So we have new antiviral drugs, but first we need to be able to tell what the virus infection is and whether these drugs would work for them. Second, technological developments, particularly in the area of nucleic acid chemistry, as I have just been showing you, have provided important new tools for diagnosis, viral diagnosis. Third, the number of patients at risk accruing opportunistic viral infections has expanded greatly as a result of HIV/AIDS epidemic. And finally, modern management of HIV infection and hepatitis C is providing a new paradigm for the integration of molecular techniques into management of chronic viral infections. What does it mean? Well, people who have HIV undergo therapy, multi-drug therapy. How do they know that it's working? By doing molecular diagnostic testing, finding out if the titer of HIV is going down or going up. Same thing with Hep C. So all these developments are not only increasing the use of diagnostic virology techniques, but have enabled the detection of a variety of emerging pathogens, such as SARS-CoV-2, that pose a threat to mankind. That's it. Thank you for listening. And any questions? Let me see.
All right, Dr. Nadala, thank you very much for that very informative presentation about diagnostic virology. I'm sure that our participants were enlightened and gained a lot from this lecture and are eager to ask their questions. So let us now proceed to the Q&A portion. For our Zoom participants, please key in your questions in our chat box. While for our Facebook and YouTube participants, you may type in your questions in the comments section so that our moderators can pick them up and be answered by Dr. Nadala.
I think they understood everything. No questions.
Maybe they're still typing in their questions. Let's just wait for a while.
All right. Maybe you have a question.
Okay, we already have here one question from Miss Baby Jane from our Zoom. She is asking, Doc, bakit po expensive ang RT-PCR test?
Yeah. Okay. Kasi yung RT-PCR test, it consists of sample preparation and the RT-PCR itself. So in sample preparation pa lang, you require a series of reagents there that you, like a kit. And you buy that from one supplier. Then yung RT-PCR naman, another supplier. And these things, before they get to you or to the labs that use it, they pass through several middlemen. Okay? They can take the cut from one place to the next. So by the time it's used in testing, and the people who use it for testing, they also need trained technicians to do the sample preparation, to run the test. And so it's a whole operation. So patong lahat yung cost. Okay. So even if a PCR test should cost, in my experience anyway, should be costing maybe 200 pesos, 300 pesos. Nagiging 2,000, 3,000 dahil sa lahat ng mga patong, plus babayaran mo pa yung gagawa ng test. Yeah? So yun ang dahilan. Reagent-wise, we can make these tests less than 200 pesos each test. Kaya lang they have to be packaged, they have to be transported from whoever made them, imported, and people along the way, yung mga distributor, they have to get their cut. And then the people who perform the test, they have to get their cut. They pay their people and get profit. So yun ang nagiging ano. So sana kung sa Pilipinas, sa atin for example, in government, meron tayong gumagawa nitong mga test na to, matatanggal lahat yan, mga middlemen. And then if we make the test easy to use, like they can be, dapat ma-automate na lahat yung mga sample prep na hindi mo kailangan ng maraming technician ang gumawa. Then, you really cut down on the price.
All right, thank you for that, Dr. Nadala. We also have a question here from Facebook from Mr. Efren Alvarado Candia. Doc, how does false positive happen in antigen tests?
Kasi yung antigen test, dahil yan sa kumakapit yung antibody, di ba? Pinakita ko na sa inyo kanina yung lateral flow test where yung virus antigen magbabind yan sa antibody tapos makakapture ng antibody doon sa test line. Yun yung nakikitang linya. So pagka hindi specific yung pagbind nila, di may linya ka pero wala naman palang antigen. In certain instances, yung for example, yung antibody na ginamit, hindi naman talaga ganun ka-specific to sa virus, hindi maganda ang kanyang specificity, di kahit na walang virus, minsan dumidikit siya doon sa linya. Ganun. O kaya yung sample, for example, yung sample hindi masyado na-extract ng mabuti or maraming mucus na kailangan or maraming mucus na dumidikit non-specifically, nagkakalinya ka kahit nawala. Hindi mo nakikita yon sa real-time PCR. Yung ganong klaseng mga kumbaga failure ng test yun kasi bakit positive dapat negative, wala namang virus. So, but it's a feature of lateral flow tests. Kung maganda lang talaga ang pag-design at pagpili ng mga antibody na ginagamit at yung target mo at saka yung buffer na ginagamit mag-extract ng viral antigen, maganda naman ang specificity na it should be more than 90%, like 95%. Pero ang marami sa mga lateral flow, hindi masyadong maganda ang pag-develop nila. They just put together whatever they can and tatawagin na nilang magandang test. Kaya nga the FDA or whoever sa regulatory sa Philippines, they have to approve these tests for use. Kasi babalidate muna nila na maganda ang specificity at sensitivity bago payagang matinda. Kasi marami diyang hindi naman maganda yung test nila na develop, tinitinda pa rin nila.
Okay, thank you, Dr. Nadala. Another question from Zoom pa rin po, from John Paul Ramos. Good morning. What are your thoughts on metagenomic analysis in animals or plants to predict viruses that can cross the species barrier and cause the next pandemic? Would it be feasible and a good strategy in preventing the next pandemic? Salamat po.
Yes, absolutely. Of course, mahirap mo you will be able to identify viruses sa animals kung we're talking about pandemic sa human pandemic. Sa animals, yung mga likely maging zoonotic by using metagenomic techniques, of course. Kasi makakuha ka lang ng sample sa, let's say, bat, and then you basically sequence everything there. You will be able to find kung ano bang sequences dyan yung dapat nasa bat or kamukha nung coronavirus o kamukha nung kung anumang other viruses. So you will see those sequences and you will know how similar or different they are from known coronaviruses, for example. But it's really hard to predict how they're going to be in human beings. Depende na yon sa similarity ng mga iba-ibang proteins nila doon sa kanilang na kinocode ng kanilang genome. Pero yes, you will be able to find, that's the way to find new viruses that we've never seen before by just basically doing metagenomic sequencing. They've already done that with like laman ng ating gastrointestinal tract. They have found new viruses there, never before seen, na napakarami pala nung kopya na nandoon. So many of that virus is in there. Pero we've never seen it before kasi they're always just there and they're not really causing disease. But since they did a metagenomic sequencing, nakita nila na nandun pala.
Okay, thank you, Doc. Another question po, may virulence factor din ba ang virus? Is it because of its envelope and non-envelope type? This is from Miss Maria Ana V. Echeverry.
No, it's not virulence factor in the sense that we see it with like bacterial virulence. Of course, some virulence factors of bacteria are actually carried by bacteriophages, but that's a different story. In terms of viruses, it doesn't depend on whether it's enveloped or not. Some viruses are just enveloped, some viruses are naked. Ang virulence ng virus is when they replicate in your cells. Yeah, how are they able to lyse your cells or are they able to just coexist in your cell? Yeah, so if they tend to lyse your cells, and as I said, a lot of viral pathology is immunopathology. So we're really overreacting to the viral infection. So part of it is yung body natin ang nagkukos ng napakalaking harm. Not necessarily just the virus itself replicating or being virulent per se, but it triggers an immune response from us that goes out of hand. So for example, ang SARS-CoV-2, bakit tayo namamatay? Dahil pag naging systemic na siya, napunta sa lungs nyo, okay? At ang dami ng nag-start na siya replicate, nagre-react yung body natin, nagpapadala ng mga cells na magla-lyse ng kung ano-ano, tsaka mga cytokines. Yung mga cytokines na yan ang nag-stimulate ng immune system natin. At ang immune system natin, pag stimulated, halos kahit anong makita niyang cells dyan, nalalyze niya. Mostly virus-infected cell, but yeah, so nangyayari so much damage in our lungs, nakukulangan na tayong oxygen. And then the virus also binds to cells sa ating heart, sa ating kidney, sa ating kidney tsaka liver. So nagkakaroon ng damage yung mga vital organs natin. And that's because na-recognize ng body natin yung presence ng virus, and then it sends out all these chemicals na nag-stimulate pa ng ating immune system, yung mga lymphocytes natin, na parang we are under attack. So yung body natin parang at war. So alam nyo naman yung mga collateral damage. So yun ang nangyayari. Pero to answer the question, there is no gene necessarily the masasabi nating virulence gene. Ang virus may mga genes or may mga proteins na designed to counter yung ating immune response to them. So maybe you can call that a virulence gene, but those are really to protect itself from being attacked.
Okay, Doc, thank you. Another question from Renzel Bascarce. Doc, I don't know if it is a hoax because some companies are now creating a vaccine. Is it really possible to create a vaccine with regards to HIV since as we know, nag-i-integrate siya permanently sa genome ng host cell?
Ang kaso ng HIV, wala pang vaccine talaga sa HIV na per se na nakaka-protect. There have been some na sabi nila mayroong some protection, but nothing really came out of it. Ang pinaka news kumbaga sa science in terms of curing people from HIV is converting your own cells, T cells primarily, to be able to recognize and attack virus-infected cells. So yun, may pasyente sila na talagang na-cure daw nila yung HIV. Pero as far as vaccine, wala naman. Yung vaccine against SARS-CoV-2, mas madaling gawin. In fact, pwede natin gawin yan sa Pilipinas. Yung style ng Chinese na ginroll lang nila yan sa cell lines and then nag-purify nila yung virus mismo. Kasi tumutubo siya sa cells, sa viral cells. So, i-purify mo lang naman yun na, patayin mo, heat inactivate mo, tapos pwede mo nang i-inject sa tao. Yun kaya nga meron tayong Virofarm ba yun? At saka yun, mga killed virus lang yun. Kaya yun gawin ng Philippines pag meron na tayong Virology Institute. Pero yung mRNA, mas komplikado. Yeah. You need a lot of technology for that.
Okay, thank you. Another one from Mr. Mark Bautista of RTU. Good morning, Doc. Can you share with us your insights on the future of diagnostic virology here in the Philippines?
Yeah. Again, sa tingin ko lang way behind tayo sa molecular tests at saka sa just rapid antigen tests and antibody tests in general. Wala tayong magandang industry dyan ng kumukuha pa tayo sa Korea South Korea and sa yung iba siguro sa China. So dapat yan ang magiging task ng ating Virology Institute. Magtayo ng Diagnostic Development Unit na gagawa nitong mga iba-ibang test na dinescribe ko sa inyo sa webinar na to. Yun ang future natin. Let's start this Virology Institute, and I personally am going to help establish a Diagnostic Development Unit na magde-develop ng mga technologies on diagnostic tests, both nucleic acid, antigen, antibody tests. And then pwede nating i-technology transfer yan sa mga industry sa Pilipinas. Pero kailangan kasi ng development muna ng magandang test bago mo i-transfer doon sa mga negosyante na gustong gumawa ng mga test, diagnostic test.
Okay, thank you again, Doc. Another question from Mr. Mark Ferrer. Doc, is it costly to do research studies on phage therapy against multi-drug resistant bacteria, given we will do a lot of cross-sectional studies using a virus, specifically bacteriophage? Thank you very much po, Doc Nadala.
It's not that difficult. Ang ano kasi ng bacteriophage, find me a virus, a bacteria, and I will give you a bacteriophage for it. Halos lahat na bacteria may bacteriophage. Ngayon, yung drug resistance, multi-drug resistance, that almost doesn't matter for the bacteriophage. Kasi, for example, we're talking about E. coli. I mean, if the bacteriophage infects regular E. coli bacteria, even if that's multi-drug resistance, the bacteriophage will still infect it and kill it. So, in other words, the difficulty probably is not in being able to find a bacteriophage that will lyse a multi-drug resistant bacteria. I think the difficulty is probably do clinical trials, and demonstrate the virus and the bacteriophage efficacy, right? We talk about that with vaccines, for example. FDA is not going to let you just sell your vaccine or introduce it. You have to have some clinical trials to prove it. Now, if I have bacteriophages that I say, okay, this will kill a certain bacteria found in hospital-acquired infections that has multi-drug resistance, then where is my proof? Yeah. So if I develop something like that, it has to be used on somebody sick with multi-drug resistant bacteria. And that would be the difficulty. But developing, researching and developing bacteriophages that can act against drug-resistant bacteria is not that difficult.
All right, thank you. Another question po from Ms. Trisha May Campit. Good morning, Doc. In your experience, what is the best or preferred diagnostic tool for viruses po? Thank you.
RT-PCR or PCR. If it's a DNA virus, use PCR. If it's an RNA virus, RT-PCR. That's it. I think those are the best. If you want it fast, then you should use one of those isothermal methods like NASBA, TMA, or RPA, recombinase polymerase assay. Those are the best diagnostic tests.
Okay, thank you. So for our last question this morning, sir, good morning. If a researcher is planning to detect COVID in animals or primates, can they use the human rapid antigen kit for the detection? As well, as we all know, mahal kasi ang cost if RT-PCR ang basis for detection on animal samples. Thank you po. This is from Hero Galamgam.
No, I wouldn't use. Number one, it's not sensitive. And number two, in these animals, normally, ang viral infection is not really very high levels of virus are being shed by the animal. Kasi nga, nagkaroon na siya ng steady state, as in, natutolerate na yung virus, hindi na nagkukos ng damage sa kanya. So, I would still number one, if I can use, as we said, next generation sequencing, so metagenomics or metatranscriptomics, that's how you can really look at what's in this animal. Yeah, what's in this bat? What is it carrying? By sequencing everything, all the nucleic acids you can find in your sample from that bat, you will know all kinds of viruses that may be in there. But if you're targeting SARS-CoV-2, then you don't have to sequence. You can probably just use one of the RT-PCR tests. Now, as far as the cost, you're talking about buying or having somebody else do the detection. But if you're doing research, trying to look for virus in animals, you should have your own RT-PCR test. And you should know yourself how to extract the nucleic acid. And if you know how, it's very cheap per test. You just have to buy the enzyme, you acquire the primers, and then you have, as long as you have the PCR machine, which is no longer expensive, then you can do your research. You can try and look for the virus.
Oh, thank you.
Oh. That signals the end of it.
Yeah, I can see that there are still questions left unanswered
Okay.
sa chat box. Pero in the interest of time, sorry, please mute your microphone. Okay. Thank you. Okay. Thank you very much, Dr. Nadala, and to our participants who actively engaged in our Q&A session. I can see na marami pa po tayong questions na hindi nasasagot sa chat box, pero in the interest of time, we'll be ending our Q&A session now. But don't worry po, for the remaining questions, we'll be sending them directly to Dr. Nadala via email so that he can answer them. Now let's present the certificate of our appreciation to the speaker. Okay, please let me read this for you. This certificate of appreciation is presented to Dr. Elpidio Cesar B. Nadala Jr., Balik Scientist of the Diagnostics for Real World Limited USA, for sharing his expertise as resource person for the webinar entitled Diagnostic Virology as part of the establishment of the Virology and Vaccine Institute of the Philippines program of the Department of Science and Technology, held online on February 21, 2022 via Zoom, given this 21st day of February 2022, signed by our DOST-ITDI Director, Dr. Annabel B. Briones. Okay, now we encourage everyone to turn on their cameras to participate in our group picture taking. Okay, first panel, 1, 2, 3, smile. Okay, second panel. Okay, next. Okay. Thank you. All right, thank you for your participation. And for the continuation of this VIP webinar series, Here is the preview of our upcoming session. Okay, this session is entitled Immunoassays by Dr. Lourdes M. Nadala, to be held on February 28, 2022 from 9:30 a.m. to 12 noon. So please mark your calendars and don't forget to register. Also, please do like and follow the Facebook page of Environment and Biotechnology Division, ITDI. The webinar will be posted in our page within the week, as well as the registration link. Okay, again, thank you to all who joined our learning session for today. Please answer the evaluation form that will be flashed on your screens later to secure a copy of your certificates. This has been Red Danganan of DOST-ITDI. Happy lunch and enjoy the rest of the day.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap liwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap liwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap liwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lilipad. Ating abutin ang pangarap liwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. sa bukas ating iparana sa bayang Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin.